our previous concept regarding the DNA and chromosomes, we will come to know that the DNA is present in the nucleus and we have 46 molecules, or in the common language, we say that we have 46 strands or 46 molecules of DNA present inside our nucleus. And all these 46 DNA, for 46 molecules of DNA would be a token of our inheritance, the genetic inheritance from parents to offsprings. These are the tokens, these are the guarantee of transport of the parental characters from the parents to the offsprings. These molecules guarantee the transport. So the question may arise that how the DNA carries the information, carries the genetic information. It would be properly said as the genetic information, but how the DNA carries the information to the next generation. uh, Number one question was, in which form DNA is carrying the information. In the last three lectures, we have understood it very clearly that DNA carries the information in the form of nucleotides, and then it converts these nucleotides into messenger RNA, and messenger RNA goes to the cytoplasm and creates the protein. As a whole, we did discuss this concept in the previous three lectures. But coming to the real point, that how is this a guarantee of our inheritance? How the DNA carries the information to the next generation? What is the role of the DNA in carrying the information, and how does it occur? First of all, the DNA threads. I did tell you that DNA is in the form of threads whenever a cell is in non-dividing phase. Whenever cell is in non-dividing phase, then the non-dividing phase mean it is not performing the mitosis or it is not performing the meiosis. Then our genetic material would be in the form of the DNA threads. If I make categorize it with the help of headings, then genetic material in the non-dividing phase, it would be present in the form of the DNA threads. But if you consider another category, in the dividing phase, in the dividing phase, the DNA is not present in the form of threads. Instead, the DNA is converted from the threads into the chromosomes, into the form of chromosomes. You may say that actually it is the folded DNA. The DNA is folded into the form of the chromosome. For example, if I give you an example of traveling, if you ever travel from one place to another, what would you do? For example, if you come to the Pakistan, or if you shift from the Pakistan to the Abu Dhabi, what would you do? You take the briefcases and then you um, fold your clothes, and then you put those clothes into that suitcase. What would happen if you haphazardly just fold the clothes out of any pattern? You just roll the clothes and throw it inside the suitcase. What would happen? There would be no place left inside the suitcase, or improper transport from one place to another may damage the cloth. There must be a very proper transport, and we fold the clothes in a very proper way so that there is some space, and the cloth may not get damaged by improper folding. If you ever just leave the traveling in your cupboard, in your cloth, जो अलमारी होती है हमारी कबर्ड होती है, if you put the cloths without any proper folding, after some time you feel that it is getting damaged. Same like that, the DNA, whenever we need to transfer the DNA from one generation to the next, we always fold the DNA. And whenever we fold the DNA, it converts into the form of the chromosomes. For example, if I have this DNA, what would I do? I would fold this DNA into this form, and now this form would be called as the chromosome. All folded form of DNA would be called as the chromosome. Um, Let me tell you a bit more detail about the chromosome. The chromosome is not only the DNA, but it is the DNA as well as some folding proteins. DNA cannot fold on its own, but it takes the help of the proteins to get folded. So it means that if this is the DNA, for example, this is the folded DNA. Actually, it was a DNA thread like this. Actually, it was a DNA thread like this one. Whenever we fold the DNA, it comes into the form of a chromosome, but it can never fold without the help of the folding proteins. So you may say that the chromosome is a cluster of DNA as well as folding proteins, and now you can carry this chromosome to the next generation in the form of loose DNA threads. For example, you may ask a question that why we cannot transfer the DNA in the form of threads, in the form of molecules. What is the danger in that? What is the problem in that? So there is a very huge problem in that. Number one, if a single DNA molecule gets missed, then it would be a life hazard for the next generation. Number two, if a single DNA molecule gets damaged, gets broken, it would be a life threat. While transferring, while getting transferred from the one generation to the next, if the DNA molecule is damaged, broken, or lost, then it would be a life threat. For example, I suppose that you have seen many children suffering with the Down syndrome, and that Down syndrome is actually loss of DNA from a particular chromosome. It means that even in the form of chromosome, the DNA can be lost. So if you do not fold the chromosome, if you do not fold the DNA into the chromosome, you can guess what would happen. The DNA can get broken, the DNA can get lost, DNA can get damaged, DNA can get mutated. So it can never be transported in the form of, especially in humans, it can never be transported in the form of a complete DNA molecule, but it has to be folded in the form of chromosomes. Understood?
Yes, sir.
Okay. I did discuss a few days ago that number of DNA molecules in human cell molecules in human nucleus. If we talk about the human nucleus, then we did discuss earlier in this lecture as well that in human nucleus the number of DNA molecules is 46. Okay, then if you talk about the apes or if you talk about the chimps, then they have the 48 chromosomes, right? And after that, if you talk about some, for example, onion, because it is a plant, it is a living thing, it must have the DNA in their nucleus, so they have the 40 chromosomes. Just like that, you may count it. There is a fruit fly, also known as Drosophila melanogaster. Do not get panicked with the name. I would tell you if it is mentioned in the curriculum. If it is, and if it is not, then you do not have to bother about it. Drosophila melanogaster. It only have 8 chromosomes. So many mosquito, I think there are some organisms which have only 4 chromosomes. Some organisms have only two DNA molecules. So the number of chromosome or number of DNA molecule in a species is a distinguishing feature or character. For example, there is no organism which has the 46 chromosome as humans, or you may say that it has 46 chromosomes same as humans. Humans have 46 chromosomes, but these 46 chromosomes are different in shapes, different in sizes as compared to other organisms. So the shape, let me specify it, the shape and the number are particular to the species. For example, if I talk about the Homo sapien, then I means that an organism with 46 chromosome. If I talk about the chimp, then it has 48 chromosome. If I talk about the 8 chromosome, then it means it is Drosophila melanogaster. The shape and number of particular are particular to the species. We can recognize the species by only the number of chromosomes present in that species. For example, there are dogs having the 44 chromosomes, 50 chromosomes, etc., etc. So there can be a difference of chromosome between different species. So coming to the point, uh, if I come back to the point where we left the lecture yesterday, diploid and haploid, then I also said that diploid and haploid are very critical in understanding the concept of inheritance. Be very attentive now. Be very carefully understand the concept of diploid or haploid. For example, I can give you an example. You have three color pencils. First color pencil is black. The other color pencil is red, and the last color pencil is blue. You may have seen the markers in the hands of your teacher of these colors, red, blue, and black, or red, blue, green, or etc. All these colors. You can see set of markers. Whenever I teach, I often whenever I teach on the board, then I often have four color markers, a set of four color markers: blue, red, black, and green. Often I have I use a single set. It means that if you open my bag, and if you open the compartment of the bag in which the markers are there, then you can see I always have a complete set of these four markers in my bag, which means it is a set. For example, a teacher uses three markers: red, blue, black. This is a complete set. But sometimes, if you ask a teacher that, sir, is a single set of these three markers is enough? Then, if a teacher is like me, who very often, who uses the colors repeatedly. I do not use a single color. I never use a single color on the board. I use the color green, black, blue, red repeatedly. My board is always colorful because being a teacher of biology. So if you ask me that, sir, only a single set is enough, then I would say no. There must be a set that is present in my hand, and there must be an extra set to support the original set, which means that if I lose this set, what would happen? I can use the extra set. I can use the spare markers. If the ink of any of this is finished, then what would happen? I can use I can use one from the other set, so that I would refill the ink from the marker. Now put this imaginary imaginary you may say that imaginary situation on the condition of the chromosome. We have 46 chromosomes in our cells, in our nuclei, in our nucleus. Nucleus is singular and nuclei is a plural. I would tell you about the singular and plural as well for the attemptation of paper. So we have 46 chromosomes in the nucleus of our cells. Actually, when you want to write these 46 chromosomes, the proper way of writing these 46 chromosomes is 23 plus 23, which means that our actual set is this 23. We have an extra set of 23 similar chromosomes that has been given to us by our parents, which means this is an original set, for example, or this is an original set, and there is another extra set present in our cells. This makes our cell as diploid. How? Because when you talk about only 23 chromosomes, the set of only 23 chromosomes is known as haploid set. And the set of two similar, oh, sorry, you may say that a group of two similar sets of chromosome in a nucleus is known as or that nucleus would be known as diploid. For example, I just told you that Drosophila melanogaster is a fly that contains the 8 chromosomes. How it contains the 8 chromosome? 8 equals to 4 plus 4, which means 4 is a haploid set. Whenever the haploid set haploid nucleus gets another set from another cell, it becomes diploid. Now this is diploid. But if you go back to this situation where only 4 chromosomes are present in the cell, then that cell would be regarded as haploid. Let me make this a bit easier, then I would come back to you for questions. For example, you have studied that after meiosis the sperms are made and the egg is also there. What is the feature of egg and sperm? Both are haploid. Both are haploid, which means there are 23 chromosomes present in the sperm coming from the father, 23 chromosomes present in the egg coming from the mother. Both of these would fuse and it would create 46 chromosome, which means that these cells are haploid. Sperms and eggs are haploid. When they fertilize, they would create, what would they create after fertilization?
A diploid cell, zygote.
Zygote, which means the zygote is diploid. So this is the theory behind the concept of diploid and a haploid cell. So whenever you come to the point where someone asks that what is a haploid cell, you may clearly speak that all those cells which are containing a single set of chromosomes, that would be a haploid cell. And if it is containing two same sets, it would be called as diploid. Two same sets. Okay, is there any question?
No, sir.
Okay. Okay, so coming back to this situation, black and then red and then blue. Okay. If I have both these sets, then black is similar to this black, red is similar to this red, blue is similar to this blue. Similarly, when you talk about the human being an organism containing the 46 chromosomes in the nucleus, 23, which is made of two sets of 23, then the chromosome number 1 of this set of 23 would be similar to the chromosome number 1 of this set of 23. First of all, let me make you clear one thing. Uh, uh, I did mention that our nuclei have two sets, 23 plus 23. One of our set is given to us by the father, and one of our set is given to us by the mother. But the shape of chromosomes are similar. For example, if this black of set A is similar to this black of set B, and the red of set A similar to red of B, and the blue of A is similar to blue of B, similarly, the chromosome number 1 of this 23 would be similar to the chromosome number 1 of the other 23. Then second would be similar to second, third would be similar to third, fourth would be similar to fourth. These are, now be very attentive, this is the concept. Whenever I teach a student, this is the concept which he or she always misses. What would be the relation of these two similar chromosomes called? These two chromosomes being similar, these two would be called as homologous chromosomes. Homologous is actually a synonymous word to Similar. Synonymous word to similar. It actually means it is containing similar information. The one of 23A is containing the information similar to the one of 23B. The 2 of 23A is containing similar information to 2 of 23B, which means that 2 and 2 are homologous. 3 and 3 are homologous. 4 and 4 are homologous. Now, coming to the point, proper point, this set 23A from father, this set was given to us by our mother. The set coming from our father has the chromosome number 1 that would be containing the similar information that is given to us by our mother. So the chromosomes of father would be homologous to the chromosomes of mother. ये है homologous chromosome का concept. जी बेटा.
Sir, I had a question.
Yes.
Sir, why are both of the, like, you said that the chromosomes from the father are similar to the chromosomes from the mother. So why are they same? And what makes them similar?
Yes, there are two different questions, but the answers of both question is same. The problem is the species of human. If you talk about the concept of evolution, then the process of evolution made our chromosomes similar to each other. For example, if we talk about the chromosomes of the human, we often have 44 chromosomes and 44 plus 2. If I talk about the humans, then I can differentiate the chromosome on the basis of structure. On the basis of structure. On the basis of structure, I can differentiate the chromosome 44 and 2. Total set is 46, right? 44 chromosomes are similar. The last two can either be similar or they can differ. If they are similar, then leave it. 44 chromosomes, these are similar and they define our body. The first 44 define our body. The last pair, which is containing the two chromosomes, this is known as sex chromosomes. Sex chromosome. The sex chromosome is such a pair that it would define our gender, which means that if both are the same, then it would be creating a female, and if both are different, then it would be creating the male. Now, coming to the role of the chromosome. If the chromosomes of two humans are different, then those humans can never be regarded as Homo sapiens. Let me repeat this. If two chromosomes, for example, this one is dissimilar to this one, this 2 is dissimilar to this 2, and this 3 is dissimilar to this 3, not similar, then what would happen? The total information, the total design, the total code of the Homo sapien would change. So it means that the basic DNA of every male and female, either from the family or far from the family, the basic DNA of human is same because it defines the basic characters of Homo sapiens. If the chromosomes get changed, then it would never be called as the Homo sapien. So being a same species, being the persons of the same species, even the DNA of male and female are 99.5% similar. Even I'm 99.8% similar. There is such a small variation that defines our genders. And if you talk about a family, for example, you may say that you are living in Pakistan and someone you are living in Abu Dhabi and someone is living in China. What is the expected difference between them? Then it would be, I think, 99%, 98 point something percent same, and the remaining portion would be different. So it means the being a same species, the human must have the same chromosomes. That is why, if some chromosomes are coming from the mother and some chromosomes are coming from the father, the basic DNA would be same. There can be slight changes according to generation. According to generation. Let me tell you something that would make it a bit more clear. You know that you can possess a character of your pardada. Pardada मैं इसलिए कह रहा हूँ, you know Urdu. Or even
Yes.
the upper generation of your pardada, you can possess a feature of that person. Why? Because the chromosomes have that information in them. The chromosomes have that information, and when those information become dominant, I would tell you the concept of dominant later, but if those characters become dominant, then that feature can arise. You may know about some familial diseases. Familial means that run in the family. The diseases that run in the family, why do they run in the family? Because our basic design is same. The basic design of our DNA is same. So if a gene is defected, if a portion of DNA is defected, it can run in the family because our basic design is same. So to be a same species, male and female both would have basic design of DNA. Sorry, similar basic design of DNA. Okay.
Yes.
Okay. What was I telling you? Yes. We were talking about the homologous chromosomes and okay. So let me write it once again. 23 plus 23. And this is chromosome number 1. Suppose this is chromosome number 1 and this is chromosome number 1 of this set, 23A, 23B. If I say that these both chromosomes are similar, then it means that the DNA present on both these chromosomes would be similar. DNA is similar. Their DNA is similar. Their structure is similar. Keep this in mind. I am not saying these chromosomes are identical. I'm saying that these chromosomes are similar. Identical means 100% copy. No, there would be a slight difference. ये जो हमें बहन भाइयों में difference नजर आता है, that is because of that slight difference between the chromosome. So these are similar, not identical. Let me explain this, then it would be clear to you. This is the chromosome number 1 or 23A. And this is the chromosome of 1 of 23B. Both are homologous. It means their DNA is similar, their structure is similar. For example, if I tell you, what did I tell you about the genes?
Uh, they are present as multiple segments in the DNA.
Oh, yes, these are the multiple segments. For example, if I say that there is a gene A present on this chromosome, the DNA of this chromosome, then there can be gene B, C, D. So this DNA would be possessing the genes, and the genes I regarded those genes as A, B, C, D. These both chromosomes are similar. The DNA present on this chromosome is similar to the DNA present on this chromosome. So it means that the DNA that the genes can also be same. For example, if there is A present on the top at the same location of the chromosome number 1 of 23B, there is also A would be present. Second position is B. So the B would also be present at the number 1 of 23B. Similarly, C and D. So it means that both of these chromosomes would have similar genes because these are what? These are homologs. These both are homologs. That is why they would be possessing the similar genes coming from the mother, coming from the father. For example, if I write M here, F here, mother coming from the mother, it would have the gene A. Same chromosome from the father also would have the gene A on the top of the chromosome number 1. So a question can arise. This is a bit more technical, I know, but be with me. I will clear out your confusion later. A question may arise that similar chromosomes are known as homologous. What would be the similar gene called? Similar genes. Similar genes are also called as alleles. 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 So it means that if two chromosomes are homologous, which means similar, then their genes would be similar. So those homologous genes, those similar genes would be named as alleles. Let me ask you one thing. Did you study this chapter in your school?
Yes, sir.
What did they tell you about alleles?
So about alleles, they're the different version of the same gene.
Different versions of the same gene. But it does not tell us that there are two form of a single gene, two location. The gene is present at the two location. One is given from the father and one is given from the mother. And these similar genes would be called as alleles. Now, coming back to a very particular point. For example, if I have this chromosome that is coming from the mother, and if I have this chromosome coming from the father, and both of these chromosomes have are homologs. Both of these chromosomes are homologous, which means if there is A, there is also A. If there is B, there also can be B, and if there is C, then there also can be C. Now be very attentive. The similar genes do not mean that the gene coming from the father would be working as same as the gene coming from the mother. For example, what do genes do? What is the function of the genes?
They carry our characteristics, for example, the
Okay.
color of the eyes, skin color,
Yes.
and
Okay. Fine. Then what do the genes make to show the characters? Genes are the DNA, right? So they
yes.
create the proteins. For example, there is a protein known as insulin. You know about the function of insulin?
Sorry, sir, could you repeat your question?
You do you know about the function of insulin?
Um, it so basically it allows the glucose to enter the body's cells so they can provide the energy.
Yes, it is more accurate answer. Commonly, it is known as reducing. It has the function of reducing the amount of glucose in our blood, but actually, it allows the glucose to enter into the cell. So, insulin is a protein. Did I tell you that if a gene gets malstructured, if the structure of the gene is not proper, or if there is something bad happened to the gene, it would not create the protein, or it would create a protein that, but that protein would be of no use to us. You remember the thalassemia?
Yes.
I told you that no hemoglobin is formed during this disease. And there is another disease known as sickle cell anemia. Sickle cell anemia. In this process, the hemoglobin is made, but that hemoglobin is not properly structured. The structure of hemoglobin is distorted. For example, if the structure of hemoglobin is like this is normal hemoglobin. What would happen in anemia? It would become like this. It would become like this. There is an X, and this is a distorted X, right? So it means that if a gene gets faulty, if a gene gets damaged, if a gene gets mutated, then it would not create, or it would not create a protein. Either it would not create a protein, or it would create a protein, but that protein would be of no use to our bodies. For example, the B gene present on the chromosome of mother is uh, is for the insulin. But you come to know that the gene coming from the father is mutated. This is a mutated gene, and this is not creating the insulin. This is not creating the insulin. It means that you have at least one gene that is working properly. You have at least one gene that is working properly to create the insulin. B gene, I did mention that the B is for the formation of insulin. You have one gene of insulin you are given from your father, and one gene of the insulin you have been given by your mother. The ideally both the genes should work. Both the genes should work, and the insulin should be made up to the capacity of both genes. Ideal. This is an ideal situation, and it happens in the human. It is not an unachievable ideal condition. It is an ideal condition, but it is achievable, and it happens in many humans that both the genes are working. But I am giving you a situation that if you are provided with a gene from the mother, and that gene is creating the insulin, and you are provided the same gene with your father, but that gene is defected, then that gene would not work. That gene would not work. The protein of that gene would be of no use to us. What would happen? The gene that is creating the protein would be regarded as dominant gene, and the gene that is not creating a protein, or creating a defected protein, this gene would be regarded as recessive partner of its gene. So let me complete my sentence. What did I tell you about the relation of these two genes? What do we call the relation?
Alleles.
So it means that the allele from the chromosome of mother is dominant, and the allele from the chromosome of father is recessive. Did you understood?
Yes, sir.
Okay, so uh, I have created a base now, right? So this is a base. I am going to, okay, first of all, I'm going to upload this on the Google Classroom, which I would make after the class. But you must revise this because to understand the inheritance, you should never forget the alleles, the dominant recessive relationship. You must never forget. So, but let me tell you another thing. After looking at the alleles, don't you think that we have at least two genes for every characters? Two alleles or two genes for every characters, isn't it?
Yes.
Just like that, if I have two sets in my bag, if I cannot use one of my black markers due to lack of ink, then I can use from the another set, the black marker from the another set. This is the benefit of two sets. This is the benefit of being diploid. This is the benefit of being diploid. That if one gene is not working, the second would work. And if both are not working, you can guess the situation. If both the genes of the insulin are not working, then you can get the result. There would be diabetes mellitus since the childhood. I hope many things are clear
Yes, sir.
because actually, first of all, Zainab, if you have anything to ask, anything, if you find it ordinary, but you must ask. Once I was in my one of my class in a coaching center, and they all said me that, sir, yes, we have understood. And then after a few days, I came to know that they were just satisfying me. They was just satisfying me that, sir, okay, okay, fine, sir, okay, fine, you have talked too much, right? Like just like that. They were just satisfying me that, sir, we have understood. So there is one advice, that there is knowledge in the question, in the repetition. There is no chance that if we do not ask, we can get the knowledge up to up to the mark, up to the standard. We can never get the knowledge without asking. Number one. Number two, if you create the hesitation inside you while asking the question, then it is just like a poison to your concepts, your knowledge. So never let me go without clearing the confusion. Never let me go. Okay?
Yes, sir.
Okay. Okay. First of all, now tell me, what do you know about the difference between the mitosis and meiosis?
So, so meiosis is basically used in the production of it's used in the production of the gametes,
Mm-hmm.
the sperm and the egg cell, and mitosis creates all the other cells in our body.
Well done, well done, well done. This is a very basic but very important difference that should be understood, that the mitosis occurs in all cells of our body. There is another very important difference. There is a difference of number of chromosomes. Can you tell what is the number of chromosome between the mitosis and meiosis? What difference does the meiosis make?
So, since meiosis makes two cells that are the gametes, so they contain half number of chromosomes in each of the cells.
Hmm. What about the mitosis?
In the mitosis, um, mm,
Mitosis creates two cells after the division. The chromosome number of the two daughter cells are similar and identical to the parent cell. Parent cell mean from which these are created. Mitosis does not create any difference in the DNA in the chromosomes. It only creates two cells. But in the meiosis, the end result are four cells. All of those four cells would have half number of chromosome than the parent cell. Half number of chromosomes. And it means that if we have the 46 chromosome in the parent cell, then after the meiosis, all four cells would be possessing 23 chromosomes rather than 46 chromosomes. Clear?
yes, sir.
Okay. Do you know about the crossing over?
I'm sorry.
Crossing over during the meiosis.
No.
Okay. See, let me see a few things, then I will get back to you. Dominant recessive, homozygous, heterozygous. Homozygous, heterozygous are different. Homologous are different. And then this monohybrid inheritance, okay, monohybrid inheritance is here. Polygenic inheritance rather than single genes. Okay, understand how to interpret family pedigree. Wow. Monohybrid crosses probabilities. Understand how the sex of a person is corrected by. Beautiful. Very beautiful. Hmm. How division of diploid cell by metal produce two cells that are let me mitosis occurs. Yes, this was I was this was I wanted to found. Meiosis produce four cells, each with half number of chromosome and the result of formation of genetically different haploid gametes. Yep, this is the thing. Hmm. 46 and the haploid number is 23. Yes, we have understood this. Now, human cells, the diploid number is 46. Variation of the species, genetic, environmental. Okay. Hmm. क्रॉसिंग ओवर के बारे में बेटा एक दो चीजें ये हैं कि okay, I will draw the drawing tomorrow in the next class. Then I will be telling you about the crossing over and the difference between the chromosome that would be created after the crossing over. Because crossing over is like the most vital process that is producing the variety between the siblings. So I would be telling you in the next class, inshallah, and that would be of much help because whenever we understand the crossing over, then our next target is easily understanding the genetics, easy understanding of the pattern of inheritance. If we understand a few things like alleles, dominant, recessive, homozygous, heterozygous, and the shuffle and the crossing over, then it would be just like a very easy concept, which would be otherwise very difficult if you do not understand these basic concepts.
Sir, by crossing over, do you mean like the crossing with like the alleles from the like the alleles from the mother and those from the father, as in the Punnett square?
No. That is fertilization. That is fertilization that Punnett scares made because for guessing the probability that which sperm can fertilize which egg of the female. That is for fertilization. The
Oh,
crossing
okay.
over occurs during the meiosis, which means that it would occur before the formation of gametes. So it occurs during the meiosis.
Okay.
Okay, beta. I think it's enough. I would be sending you a link and then you can join the Google Classroom.